students, welcome back again. I am Samapti, your zoology teacher teaching in Sri Chaitanya Institute. Students, we were discussing the chapter excretory products and its elimination. We already discussed about the structure of the nephron. Today we are going to start with a very important topic, and why did I say very important? As you can see that mechanism of urine formation, which we are going to discuss right now. Questions have been asked frequently from this part. NEET 2017, 2-3 questions were there. 2016, 2012, 2010, 2011 also. So very important for the perspective of NEET. Now talking about the mechanism of urine formation, this occurs in 3 steps. One is ultrafiltration, then is reabsorption, and then is tubular secretion. So we are going to discuss these 3 steps one by one. Talking about the very first step of urine formation, which is ultrafiltration. Now when you say ultrafiltration, students, you now very well know that the filtration is taking place through 3 layers. One is the endothelium. Where is this endothelium layer is present? This endothelium is the simple squamous epithelium of glomerulus. So the endothelium of glomerulus, and then it also passes through the podocytes, if you remember. The podocytes were the cells of the Bowman's capsule. This is also modified simple squamous epithelium. And in between these 2 layers, you had a basement membrane. So the filtration membrane is formed of 3 layers, but 2 cellular layer and 1 acellular or non-cellular layer. Got it? Now, when you talk about the ultrafiltration, students, ultrafiltration is merely occurring because of the great pressure created. Afferent arteriole having the larger diameter, forming the network of capillaries, glomerulus having very small diameter. So because of this excessive pressure, blood is getting filtered out. It is not at all an active process, it's a passive and non-selective process. Now what part of blood is getting filtered out? It's the plasma part which is getting filtered out through this filtration membrane. Large size proteins, because of being larger in size, are unable to cross that filtration slit formed by the podocytes. So large size proteins won't be getting filtered out. Now talking about what percentage is coming into your kidneys that undergoes ultrafiltration, students, I have written that about 1/5 of the blood pumped by the heart, which is approximately 1100 ml to 1200 ml, this 1/5 of blood is coming in the kidneys and undergoing the process of ultrafiltration. Is it the GFR? Lot of students get confused that this is only the GFR. No, this is only the amount of blood undergoing ultrafiltration. What is this GFR then? GFR, as you can see, is glomerular filtration rate. When you say glomerular filtration rate, it is defined as the amount of filtrate formed per unit time. So per unit time, the amount of filtrate which is formed is GFR. And the value you will remember is 125 ml per minute, or it is 180 liters per day. So 125 ml per minute or 180 liter per day is the amount of filtrate formed per unit time, defining your GFR. So that's about beta ultrafiltration. Now talking about the second process, which is again quite important, is the process of reabsorption. Now students, you will see in the last part of the chapter that the amount of urine which is formed in one day is 1 to 1.5 liter. But do you see the amount of GFR? That is 180 liter per day. If the urine formed is only 1 to 1.5 liter, where is the other filtrate gone? Actually, 99% of the filtrate is getting reabsorbed from the parts of the nephron. We are going to see which part of the nephron is absorbed in absorption of what. Now talking about the very first part, PCT, which is proximal convoluted tubule. We already discussed that PCT is lined by brush bordered cuboidal epithelium. It is having microvilli that can increase the surface area for maximum reabsorption. It is also having lot of stored food, lot of mitochondria to produce energy. Talking about PCT, I have written that maximum reabsorption, students remember, maximum reabsorption is taking place from PCT. 70 to 80% electrolytes and water is getting reabsorbed from this part. Now when the electrolytes as well as water is getting reabsorbed from this part, when you see the concentration, compare it with the concentration of blood, actually the concentration of the filtrate in the PCT is isotonic. Why? Because you have absorbed equal water and electrolytes from this part, so the filtrate remains isotonic. Now talking about the active and passive reabsorption, glucose, amino acids, I have written active reabsorption from PCT. Not only this, some ions like sodium ions is also getting actively reabsorbed from the parts of PCT. So glucose, amino acids, active reabsorption. Also talking about some nitrogenous waste also get reabsorbed, but this reabsorption is quite passive. I have written that some amount of urea is getting passively reabsorbed from the parts of PCT. Students, just remember one point, that whatsoever is the condition, maximum reabsorption occurs from the part of PCT. Talking about the loop of Henle, which is the next part, let us see which things are getting reabsorbed from the loop of Henle. Talking about the descending loop of Henle, I have written an important point that it is permeable only for water and very least permeable for electrolytes. Now talking about the descending loop of Henle, which is maximally permeable for water, as the filtrate is moving downwards, what you are reabsorbing? You are reabsorbing all the water. When you reabsorb all the water and the filtrate moves down, you will see that the filtrate is becoming hypertonic. Hypertonic means more concentrated. In fact, students, at the base, at the loop of Henle's base, you will see the maximum concentration, that is 1200 milliosmol per liter. That's the maximum concentration that you see at the base of the loop of Henle. Also students, if I talk about the loop of Henle is the part, overall loop of Henle we say is the part which is responsible for concentrating the urine, which is responsible for concentrating the filtrate. I have written here only in the descending loop of Henle, but overall if you are asked that which part of the nephron is responsible for concentrating the filtrate, that would be your loop of Henle. You can easily relate it with the animals which do not need to conserve a lot of water, who are living in the atmosphere where the environment where a lot of water is there. Their loop of Henle is absent either or very reduced because they don't need any concentration, but we need to concentrate the urine because we need to conserve water. So the loop of Henle plays a very important role, the most important role in concentrating the filtrate. Now talking about ascending loop of Henle, exactly opposite to descending. Descending was permeable only for water, least permeable for electrolytes. The ascending loop of Henle is permeable only for electrolytes, and we see that as the filtrate is moving in the upward direction, you are reabsorbing the electrolytes. Now electrolytes may get absorbed actively as well as passively because ascending loop of Henle has two parts, the thin and the thick. So in the thin region, what you see is the passive reabsorption, whereas in the thick portion, you see the sodium potassium active pump. So there is an active reabsorption. So even active and passive reabsorption can take place, but once you are moving up and you are reabsorbing all the electrolytes, the filtrate is becoming hypotonic. So here hypotonic and here hypertonic. Alright, I have also written one point that ascending loop of Henle shows minimum reabsorption. Reabsorption is minimum. Now talking about the next part, which is DCT, conditional reabsorption takes place. What is taking place? Conditional reabsorption. Now what is this conditional reabsorption? Students, we have very well regulation of the kidney function. Whenever we see there is a loss of more body fluids or there is too much of accumulation of body fluids, we can adjust that and we can adjust that using some controls. So in the DCT also, two hormones can act like aldosterone, which is a mineralocorticoid, and ADH or vasopressin. Whenever ADH or vasopressin is acting on the DCT, you are reabsorbing water. So it's conditional. When there is an excessive loss of water, that's the condition when your ADH is going to act upon the DCT and water is going to be reabsorbed. Talking about aldosterone, when aldosterone acts upon DCT students, aldosterone, since it is a mineralocorticoid, so first of all what it is going to do is the reabsorption of mineral sodium ions. When the minerals are getting reabsorbed, water is ultimately moving passively. Got it? So conditional reabsorption from DCT. Also I have written a very important point, students, about bicarbonate. In your NCERT it is given that HCO3 minus bicarbonate is absorbed. Means first time in PCT, absorbed from PCT but reabsorbed from DCT. Absorbed very first time from PCT, reabsorbed from DCT. Talking about collecting duct, which is the last part, large amount of water is getting reabsorbed from the collecting duct. Also, some amount of urea is getting reabsorbed. This urea moves into the space, that is the interstitium, and maintains the high concentration of the medullary interstitium. We will be discussing about that urea in counter current mechanism. So that's the second step in the urine formation, which is reabsorption. The third step in formation of urine is tubular secretion. The major function of this tubular secretion is to maintain the pH and ionic balance. Now students, there is a difference that is given in the theory of NCERT and in the diagram. But remember that the diagram is more correct. We will see that which ions are secreted from which part. Talking about PCT, the ammonium ions and water is being secreted, and talking about secretion means where are you secreting this? Secretion means you are secreting, you are putting everything into the filtrate, right? If suppose this is the region of DCT, okay, and filtrate is present inside, whatever arrow is made from the filtrate, is reabsorption. But made towards the filtrate, if you make the arrow towards the filtrate, that will be your secretion. So ammonium ions and hydrogen ions in the region of PCT, but hydrogen ions and potassium ions in the region of DCT as well as collecting duct. Do you get which ions are secreted in which part? Now looking at an ion which is common to all, hydrogen ions. This is the reason why so much of hydrogen ion is present in your urine. Urine is very much acidic. Now talking about this point which I have written, students, human produces nearly 4 times concentrated urine. So we have the ability of producing from the initial filtrate, if you compare, we can produce 4 times concentrated urine, right? Because we have reabsorbed all the water. Now talking about a very important mechanism, which is counter current mechanism. Now which part plays a very important role in counter current is loop of Henle and vasa recta. Talking about counter current and why do you call it as counter current? So I have made a rough idea diagram showing the regions of cortex and medulla first. Now consider this outer part as cortex, where I have made this PCT and I have made this DCT. That's the outer region, cortex, having the concentration 300 milliosmol per liter. As you move inside where your loop of Henle is penetrating, that area is called as the medullary region. This is the innermost medulla where you see maximum concentration of 1200 milliosmol. Starting from cortex 300, moving inside to 1200 milliosmol. Now this is the descending loop of Henle, and you know that the movement of filtrate in the descending loop of Henle is in downward direction. And why it is called counter current is because when you look at the movement of filtrate in the ascending loop of Henle, that's in the upward direction. So descending loop of Henle, downward direction, ascending loop of Henle, upward direction, forming a counter current. Not only this, the vasa recta, which runs parallel with this loop of Henle, the vasa recta, students, that run parallel with the ascending loop of Henle is called as descending vasa recta, obviously. Can you look at the flow of the blood in the downward direction? And the filtrate and the vasa recta that moves parallel with the descending loop of Henle is called as ascending vasa recta. Got it? Parallel with descending loop of Henle is ascending vasa recta. Parallel with ascending loop of Henle is descending vasa recta. The flow of blood in the ascending and descending vasa recta is also in the opposite direction, thus forming counter current. Now see how this counter current mechanism is helping in concentrating the urine. Now students, quite easy to remember, follow what I have written, okay? You now know that the descending loop of Henle is permeable only for water. It was mostly for water, right? So what you see that the water is moving out of this descending loop of Henle. And this water which is being reabsorbed from the descending loop of Henle, do you see where it is moving? It is moving in the ascending loop of ascending vasa recta, which runs parallel with the descending loop of Henle. Whereas the electrolyte that you reabsorbed from ascending loop of Henle, that is moving where? That is moving in the descending vasa recta. So whatever you have reabsorbed from the descending loop of Henle, you pass it to the ascending vasa recta, and whatever you have reabsorbed from the ascending loop of Henle, you pass it to the descending vasa recta. That's very much easy. So water moving into the ascending vasa recta, NaCl moving into the descending vasa recta. Now the vasa recta which took all the water, that was your ascending vasa recta, is releasing a very important electrolyte, NaCl, in the space, in the interstitium. This NaCl is very much important in maintaining the high osmolarity of the interstitium. One of the electrolytes which is very important in maintaining the high osmolarity of NaCl of the interstitium is NaCl. Where is this NaCl coming from? From the ascending vasa recta. Got it? Now this vasa recta which was taking all the NaCl from the ascending loop of Henle is releasing some amount of water in the interstitium. Do you see that there is opposite release of things than whatever was taken by them? The ascending loop or ascending vasa recta was taking water, releasing NaCl. The descending vasa recta taking NaCl, releasing water. Got it? But not only this, NaCl is maintaining the high osmolarity of interstitium. If you remember, collecting duct was also taking some urea, reabsorbing some urea. Some of the urea is also maintained in your interstitium to maintain the high osmolarity, and some of the urea is again passing through the ascending loop of Henle and continuing the cycle. Got it? So urea along with NaCl is responsible for maintaining the high osmolarity of the medullary interstitium, and very easy to know that how the counter current mechanism is helping in the concentration of urine. Also students, remember that the concentration of the interstitium increases from 300 to 1200 milliosmol. And also the concentration of filtrate, you know that starting from PCT is also lowest 300 milliosmol, and at the base of the loop of Henle we discussed that it is 1200 milliosmol, highest in the medullary interstitium, lowest in the cortex area. Now students, let's talk about another very important topic, which is regulation of kidney functions. Questions are asked from this topic also 2012, 2014. In fact, related questions are asked regarding this topic. I understand that you get confused in the regulation of kidney function, but today you will be clear in all the regulations if you listen carefully whatever I am saying. So we are going to discuss the three methods of regulation of kidney function: by hypothalamus, by RAS, and by your heart. Okay, these are the three mechanisms. Now students, remember that whenever there is a change in the concentration of the body fluid, whenever there is a change in the blood volume, we can control it by the feedback mechanisms. There are certain osmoreceptors in your body which get stimulated by change in the ionic concentration or by change in the blood volume. Now, when upon this activation of the osmoreceptors, whatever changes have been taken place in your body can be brought back to normal. Now, talking about one of such regulations is the regulation by your hypothalamus. Now, students, two things you will remember. One is in which condition the regulation is taking place, and second, that what it is responsible for doing. Okay. First thing is more important, that when this thing is going to be operated. Now, talking about the regulation by hypothalamus, students, hypothalamus is operating in response to low GFR. Remember, when it is operating during low GFR. So, ultimate function will be to increase the GFR. Am I clear? Now, when you are saying that it is operating in low GFR, why this low GFR is there? We said that whenever the GFR is low, that means the amount of filtrate formed is low, right? Amount of filtrate which is formed is less. Now, when I am saying that the amount of filtrate is less, why the amount of filtrate is less? Because maybe the amount of blood that was coming, that was undergoing ultrafiltration is less. That means if the amount of blood coming undergoing ultrafiltration is less, that means your blood volume is less. And when your blood volume is less, that means you need to understand that your plasma, your water is less. That means you will be operating all the processes that is going to reabsorb more water. Did you get my point? So it is operating when the GFR is low. Now, when the GFR is low, these osmoreceptors get stimulated by the reduction in the GFR. Upon activation of the osmoreceptors, that signals which part? That signals hypothalamus. When the hypothalamus is getting those signals, hypothalamus starts to release the ADH, anti-diuretic hormone, also called as vasopressin. It starts releasing the ADH. ADH is stored in which part? Posterior pituitary. So, it starts releasing this ADH from posterior pituitary, and we have already discussed that this ADH acts upon DCT from where conditional reabsorption was taking place. So, when this ADH or vasopressin acts upon DCT, it is responsible for reabsorbing a lot of water. And when you are reabsorbing a lot of water, that water is passed to your blood and your blood volume is increasing. That was your purpose, to increase the blood volume. When your blood volume is increased, ultimately your GFR will be increased. That was the purpose, because we were operating this in response to low GFR. Got it? Exactly like hypothalamus, which is operating during the low GFR, we have another mechanism that also works in response to low GFR only, and that is called as RAS. RAAS, which stands for renin angiotensin aldosterone system. Now remember, the spelling of this renin is not double N. Double N for the proteinaceous protein digesting enzyme by the stomach, right? This is single N, this is hormone. So why is it called as renin angiotensin aldosterone system? Is because you will see the involvement of all of this together. Now again, students, very important, I told you to remember that it is operating in response to low GFR, just like the hypothalamus. So when the GFR is low, that means you need to do all the mechanisms to reabsorb more and more water. Correct? Now what is happening here? Where is this renin produced from? I told you that the JG cells of afferent arteriole is responsible for production of renin. But how did it know that I need to start producing renin? I told you that GFR is low. When the GFR is low, then the amount of blood that is undergoing ultrafiltration is also less. So afferent arteriole, the amount of blood in the afferent arteriole will be less. In response to which, detected by the JG cells start releasing the renin. And this renin is responsible for conversion of angiotensinogen into angiotensin 1. Remember, this angiotensinogen is produced in the liver. So this renin is converting this angiotensinogen into angiotensin 1. This angiotensin 1 is getting further converted into angiotensin 2, and this conversion is under the presence of the hormone ACE, which is angiotensin converting enzyme in the lungs. Got it? So GFR was low, renin is produced by the JG cells, converting the angiotensinogen into angiotensin 1. The angiotensin 1 is getting converted into angiotensin 2 in response to ACE. So we saw the involvement of two things, that is renin and angiotensin. Now all the functions will be performed by this angiotensin 2. Now look at the function of this angiotensin 2. Number one, it can direct the proximal convoluted tubule. It can directly act upon PCT and it can guide the PCT to reabsorb more NaCl and more water. And when more water is reabsorbed, the GFR will be increased. That was our purpose. Talking about the second function, which is quite important, it can direct the adrenal cortex. Angiotensin 2 is acting upon the adrenal cortex and it is guiding the adrenal cortex to produce aldosterone. This aldosterone is a mineralocorticoid, and I already discussed that upon DCT, when the aldosterone is going to act, this is going to cause the reabsorption of Na plus and the water is going to be passively reabsorbed. Again, GFR will be increased, which was our purpose. And the last thing is the angiotensin 2 can act upon the efferent arteriole, students. Now why it is not acting upon afferent and why it is acting upon efferent? Afferent arteriole brings the blood. Efferent arteriole carries away the blood. If you are constricting the arteriole which carries away the blood, suppose I constrict the efferent arteriole, what you are doing is preventing the blood to go out of the glomerulus, right? If I constrict the efferent arteriole, that means blood will be retained in the glomerulus. If the blood is retained in the glomerulus, the GFR will be increased. That was our purpose because it was operating in response to low GFR. Talking about the regulation by the heart. Now students, remember that heart is operating in response to increase in GFR. Hypothalamus in response to low GFR. The RAS in response to low GFR, but heart is operating in response to increased GFR. Now what happens during increased GFR? That means your blood volume is already increased. You will stop everything that was increasing your blood volume, right? So what is happening, that in response to that, atrial wall, atria of the heart starts producing the factor ANF, which is atrial natriuretic factor. Now I will tell you why is it called as natriuretic factor. The very first thing you will remember that, of course, this ANF has to check upon the RAS. RAS was responsible for increasing the GFR, but you need to check upon the RAS because already the GFR is increased. How it is keeping a check over the RAS? It is inhibiting renin. It is not allowing the renin to be released, inhibiting the renin. It is also inhibiting aldosterone because both were responsible for increasing the GFR. Also students, when you have no aldosterone, that means you are not reabsorbing Na plus. That means more Na plus is releasing out of your urine, and that's the reason why it is called as natriuretic factor. Natri means sodium ions, and sodium ions are seen more in the urine, hence a natriuretic factor. This is the only factor, ANF, that is responsible for vasodilation. That's how it is helping in the bringing back the GFR to normal. So that was the regulation of kidney function. Now talking about the mechanism of urine formation, not the urine formation, mechanism of urination, which is called as micturition, we are going to see how we are passing the urine. This is very well understood by the neural mechanisms. You have the control of your nervous system for the urination process. called as the micturition reflex. Now what is happening? I have made a flow chart regarding this. All right. Now talking about very first step, that your urinary bladder is getting filled with the urine. Now once your urinary bladder is getting filled with the urine, what is happening is stretching of the urinary bladder, right? There is a stretching of the urinary bladder. Why there is a stretching? And the stretch receptors in response of this filling of the urinary bladder is giving a signal to your central nervous system. See, I have given filling of the urinary bladder, these stretch receptors are activated. Where these stretch receptors are located? In the wall of urinary bladder. Upon giving signal to your central nervous system, the action is performed by a part of autonomic nervous system, which is parasympathetic nervous system. Now when your parasympathetic nervous system is functioning, it is acting on the smooth muscles. You remember what were the smooth muscles of urinary bladder? That was detrusor muscles. When your parasympathetic nervous system is acting upon the urinary bladder, contraction of the smooth muscles takes place. Then the contraction of the urinary bladder is taking, your urination is performed. But contraction of muscles is occurring, but the sphincters which are located in the neck region, they are getting relaxed. Involuntary relaxation of the internal sphincter and voluntary relaxation of the external sphincter causes this urination or micturition. So that's the steps involved in the urine formation. Now once your urine is released, let us see what is the composition of the urine. We said that 99% of the filtrate is getting reabsorbed. Only 1 to 1.5 liter of urine is formed every day, being very much acidic, approximately 6, because we saw that hydrogen ions were secreted into the filtrate. There is a specific yellowish color of the urine, which is because of the pigment urochrome, a derivative of bilirubin and biliverdin. Also we excrete out a large amount of urea, which is 25 to 30 gram per day. So that's the composition of the urine. Also students, remember that if you analyze the urine, the abnormal constituents of the urine can help you in determining some of the metabolic disorders. Say for example, if you see presence of large amount of glucose in the urine, that is glycosuria, or presence of large amount of ketone bodies in your urine, which is ketonuria, can be indicative of a disorder, diabetes mellitus. Also students, for example, if the proteins are getting filtered out and coming out of urine in large quantities, that also tells you that maybe some problem is there in the filtration membrane, that is glomerulonephritis. It is one of the disorders that is given in the excretory system in the last part in your NCERT. Also there can be lot more other conditions, abnormal conditions of the urine, like excessive loss of water through urine is also indicative of that your ADH is not working. And when there is excessive loss of water through the urine, that is indicative of another type of diabetes, which is called as diabetes insipidus, right? That is in hormones also chapters we will discuss. Anyway, now talking about role of some other organs in excretion students, we have got lungs, and lungs can also help in excretion of large amount of water vapor as well as CO2. So there is a information given in your NCERT that your lungs can remove 200 ml of CO2 per minute. Also remember students, I have not mentioned the questions, but this topic, the role of other excretory organs, also questions have been asked in NEET. Talking about the other organ, which is the liver, liver is the largest gland in your body, which can also excrete out some amount of cholesterol, degraded steroids, vitamins, drugs, bilirubin and biliverdin. So it can also help in excretion of cholesterol, degraded steroids, vitamins, bilirubin, biliverdin, some drugs. Also helping in excretion. Talking about skin, which is the largest organ, it has got sweat glands, it has got sebaceous gland. The product of the sweat gland is sweat and the product of sebaceous gland is sebum. What is the composition of this sweat? It has NaCl, it has urea, it has lactic acid, it has water. So remember the constituents of the sweat. Also the sebum, which is the secretion of the sebaceous gland, consists of sterols, it consists of waxes, it consists of hydrocarbons. So these are the role of the other excretory organs in excretion. The last part of this chapter is the disorders. The last part of this chapter is the disorders of the excretory system. So questions in NEET 2018, there was a match the following asked from the disorders, so quite easy. We have already discussed one of the disorder, which is glomerulonephritis, that is the inflammation in the glomerular filtration membrane, due to which proteins also get filtered out. Talking about a disorder which is uremia. Uremia is a condition where your urea is retained in the blood. Urea is in the blood. So what will happen if all the excretory product is getting accumulated in the blood? That means your kidney is not functioning properly. Now to correct the condition of uremia in the patient, what is performed is called as hemodialysis. Now let's look at the hemodialysis. Let's take a rough idea. This is not a proper diagram. This is to get a rough idea about how the hemodialysis is performed. Now looking at the patient students, patient blood is having high amount of urea which you want to remove. You have taken the blood from a convenient artery, which is usually the radial artery, okay, in hands. So using a convenient artery, you have taken all the blood. And you brought the blood to 0 degree Celsius temperature so that no enzymatic reaction is taking place. So first it is brought to 0 degree Celsius temperature. Then to prevent any kind of coagulation, what you have added is the anticoagulant, so that no coagulation is taking place. Now you are passing that blood to an artificial kidney setup. How the setup is there? There is a tube through which you pass the blood having high amount of urea. That tube is called as cellophane tube. Now when you pass the blood through the cellophane tube is in contact with some solution which is called as dialyzing solution. Now you need to remember the composition of the dialyzing solution. I have mentioned that the composition is similar to blood plasma except the nitrogenous waste. The dialyzing solution has everything except the nitrogenous waste. Now why is it so, students? So that when you move the blood through this tube, this has nitrogenous waste, this has high amount of urea. This nitrogenous waste can passively move in the dialyzing solution because the dialyzing solution has no nitrogenous waste. But to prevent the movement of any other important proteins, the protein composition of the dialyzing solution is exactly like that of your blood plasma, so that does not come out. So what you see is when you pass the blood through this part, all your toxic product is moving in your dialyzing solution. From one end, fresh dialyzing solution is added. From other end, the used dialyzing solution is being removed. Once your blood is filtered, once your blood is clear, now you can return the blood to your body by bringing it to a body temperature, by adding the coagulant through the convenient vein, okay? Through the convenient vein, you are returning that into your body. That's how hemodialysis is performed. Alright, talking about the another disorder which is renal calculi. So when you say renal calculi, calculi is basically the formation of stones. If the kidney shows the formation of stones, stones can be formed of any salts, right? The crystallized mass of insoluble salts is called as a renal calculi. I have written like the calcium carbonate salts, any salt, calcium oxalate salts, if they get insolubilized as masses, that is called as renal calculi. Now students, there is a term given as kidney transplantation. Now when this kidney transplantation is performed, actually whenever you see there is an acute renal failure, in that case you need to transplant the kidney. Now whenever the kidney transplantation is done, always the kidney is taken from a preferable donor having the maximum matching. So organ is matched before kidney transplantation during the acute renal failure, and also matching is done, but there is a chance that the receiver may show some rejection. And in the immunity chapter, you know that cell-mediated immune response is responsible for this rejection, right? Also to prevent any kind of, unwanted immune response generation, often immunosuppressants are given to the patient who is receiving the kidney. And these immunosuppressants are like cyclosporin may be given. Do you know any immunosuppressant sporin? Do you know any other immunosuppressant hormone that you have studied? Cortisol is also a very good immunosuppressant. So cortisol, cyclosporin may be given to prevent the unwanted immune response generation during the kidney transplantation performed during acute renal failure. So these are some of the disorders that was discussed in your NCERT. Excretion students, remember that this is one of the chapter which is purely asked from NCERT. So try to focus on NCERT line by line, try to follow the notes and try to give good examination. Till then, we are done with this chapter. Take care, prepare well, stay happy, bye bye.